Hello everyone. In this video we're going to be talking about biochemical signaling, and we'll specifically talk about the adenylate cyclase signaling pathway, right? Adenylate cyclase is a transmembrane protein that produces cyclic AMP, which binds to protein kinase A, activates proteins through phosphorylation, which activates in turn a number of different cellular proteins. Okay. And adenylate cyclase itself is activated by heterotrimeric G proteins, right? It's a class of protein known as heterotrimeric G proteins. They bind to specific receptors within the membrane, and their binding to the receptor is really triggered by a signaling molecule binding to the receptor. And such receptors are known as Gprotein-coupled receptor. So once a hormone or a signaling molecule binds to the receptor will undergo conformational changes within the cytoplasm, which in turn will activate the heterotrimeric G protein, right? It will activate it, it will trigger its binding to the receptor, or the Gprotein-coupled receptor, and once it binds and it activates, it's heterotrimeric, so one of its three subunits will dissociate and will in turn activate or stimulate, in this case, the activation of adenylate cyclase, which we'll talk about, right? And in general, those heterotrimeric G protein have similar characteristics, and they have similar structural motifs, and all of which also have a guanine nucleotide binding domain, both for GTP and GDP. And we'll take a look at how the heterotrimeric G protein is activated when bound to GTP, and when bound to GDP, it is inactivated. All right. So we'll talk about we'll talk about all of those really in detail in a minute. All right, so the specific signaling pathway that we'll talk about activates adenylate cyclase, or the heterotrimeric G protein will stimulate the activation of adenylate cyclase. It is a transmembrane protein. We'll talk about its structure its structural details in a second, but it has nucleotide binding domains. It specifically binds 4 different ATP molecules, and it catalyzes once it's activated, once adenylate cyclase is activated by one of the subunits of a heterotrimeric G protein. It will catalyze the synthesis of a secondary messenger cyclic AMP from ATP. So adenylate cyclase is basically the enzyme, right? It binds ATP, and once the protein is activated, it will catalyze the hydrolysis of ATP to form cyclic AMP molecule, right? And we'll see how cyclic AMP acts as a secondary messenger, activating protein kinases, proteins that are responsible for the phosphorylation of other proteins. Okay, so a brief example of how a heterotrimeric G protein signal will go through. If you have a specific hormone or a signaling molecule, and it binds to its receptor, and its receptor is basically a G protein coupled receptor, right? Meaning that once that signaling molecule binds to the receptor itself will undergo conformational changes within the cytoplasmic end of that membrane, which in turn will trigger G protein, right, to displace its bound GDP by GTP, right? So now this G protein becomes active, which in turn catalyzes. All right, so the replacements of GDP by GTP. And notice those G proteins are anchored to the membrane, typically through N-glycosidic protein or through a specific palmitoyl residue that is linked to a cysteine within the protein. Meanwhile, once it's active, it will separate, one of its three subunits will separate, and it will bind to, in this case, adenylate cyclase, activating it. Well, once it's active, its ATP molecules are converted into cyclic AMP, which will trigger, a number of different cellular responses by activating a number of different cellular proteins through kinases, right? The hydrolysis of its bound GTP back to GDP inactivates the protein, right? Which in turn will basically turn off that signaling cascade produced by adenylate cyclase. All right, so a signaling molecule binds to the receptor. The receptor undergoes conformational changes, which triggers a G protein to bind to displace its bound GDP by GTP, binding to the receptor, and then diffusing through the membrane, right? Binding to adenylate cyclase, activating it. Okay, so we're going to take a look at Gprotein coupled receptors, which are a target for therapeutic drugs. And that's because they include a number of different receptors for nucleosides, for nucleotides, for calcium, and for a different number of peptides and hormones, protein hormones as well, right? And there's about 3.5% of the genes in humans are basically Gprotein coupled receptors. One example is the human beta adrenergic receptor, and it is shown bound to carazolol in lieu of epinephrine. And the ligand binding site is really within the within the membrane itself. It is nearby the alpha helices that are that make up the receptor, but part of it is also interacting with the portion of the protein that is exposed to the extracellular surface. And if you notice, the protein itself is anchored to the membrane, right, through a palmitoyl group as well, linked to a specific cysteine residue. All right, so we'll notice that ligand binding site are within the membrane itself, right? And those Gprotein coupled receptors typically only bind a small number of ligands. They're specific to specific ligands. Okay, and the ligand binding site is within the membrane. All right, so we're going to take a look at adenylate cyclase that is, again, activated by that specific hormone or signaling molecule that will bind to the receptor, right? And the receptor will activate the G protein, and the G protein will in turn activate adenylate cyclase, right? And adenylate cyclase is a transmembrane glycoprotein. It is composed of an N-terminal domain, a C-terminal domain, and transmembrane domain, which are labeled as the M domains, and it has catalytic domains that are labeled as C1a, C1b, C2a, and C2b, right? Some of those C domain, the cytoplasmic domains, are catalytic domains, and some are regulatory domains, meaning they bind regulatory molecules. And we'll see in a minute that adenylate cyclase is activated by one of the three subunits of the G protein binding to adenylate cyclase, and that is the alpha subunit of the G protein. It will bind to the C1a unit activating adenylate cyclase, right? And once it's activated, it has three different ATP molecules within its, one of its C2a subunit that converts its bound ATP to cyclic AMP. Okay, it is also can be inhibited and activated by binding to a number of different molecules. It can be activated by calcium, by protein kinase A, by calmodulin bound to calcium, which come on different signaling pathways. Meanwhile, adenylate cyclase activates protein kinases, and kinases are enzymes that catalyze the phosphorylation of other proteins, thereby activating it. Protein kinase A is a cyclic AMP-dependent protein kinase, meaning that it is activated by binding to the secondary messenger cyclic AMP, and it recognizes specific sequences where you have arginine residues followed by a small residue and either a serine or a threonine that will become phosphorylated, and then Y typically is a hydrophobic residue, right? So it is activated by binding to cyclic AMP. In the absence of cyclic AMP, that protein kinase A is inactive, and hence it is a cyclic AMP-dependent protein kinase. So cyclic AMP must be bound to the protein when the protein is, for the protein to be in its active conformation. And what we'll take a look is at the detailed structure of protein kinase A. Right? It has 2 regulatory subunits and 2 catalytic subunits, right? So it has 2 regulatory subunits and 2 catalytic subunits. And cyclic AMP specifically binds to the regulatory subunits, activating the protein, and we'll see how, right? Actually tells you how right here. When the cyclic AMP binds to the regulatory subunit, that causes the catalytic subunits to dissociate from the protein, right? Which activates protein kinase A. So we're going to take a look at the moment of one of the catalytic subunits of protein kinase A. It is showing us the N-terminal domain, which is composed mainly of anti-parallel beta sheets. It's also composed of a short helix, alpha helix and a longer alpha helix, whereas the C-terminal domain is mainly composed of alpha helices, right? Now, in the ATP binding domain, it is shown right here, and it binds to the N-terminal domain, whereas within the C-terminal domain of the catalytic subunit, that's where the peptide or the target sequence that will become phosphorylated is bound, right? So it is shown right here, but it's shown as an inhibitor sequence where A really would have been really the threonine or the serine that is being phosphorylated, but in this case it has alanine instead, right? And remember, it recognizes sequences where you have 2 arginine, a small molecule, and then serine or threonine, and then typically a hydrophobic residue. Right? And near that target sequence, there's this activation loop, which is shown in blue. And that has a phosphorylated threonine residue. And nearby the active site is an arginine residue that stabilizes those charges from the phosphate. And there is also an aspartic acid residue that is essential for the activation of the target protein. Right? So if you notice, that sequence within the target protein is close to the gamma phosphate group of ATP. All right. And shown here is the regulatory subunit of protein kinase A, and it has 2 different domains. Now, cyclic AMP specifically binds, we said, to the regulatory subunits, and there's 2 cyclic AMPs that bind per regulatory subunit. So the N-terminal domain is shown as cyan. The C-terminal domain of that regulatory subunit is shown in gold, orange, and there is a cyclic AMP binding site on either domain. And there is a specific segment of the regulatory subunit known as the autoinhibitor segment, which is shown in the magenta color right here. All right. That autoinhibitor segment fits right within the active site, or right within the substrate binding site of the catalytic subunit, which prevents the substrate that will become phosphorylated from binding. So what happens is when cyclic AMP now activates protein kinase A, and it does so by binding cyclic AMP molecule within the C-terminal domain of the regulatory subunit. Once that particular cyclic AMP molecule is bound, the regulatory subunit undergoes conformational changes, which allows another cyclic AMP molecule to bind to the N-terminal domain of the same regulatory subunit, which then in turn releases that autoinhibitor segment from the catalytic subunit or from the active site of the catalytic subunit, and then the substrate can bind, right? Okay. So how is the magnitude of a specific signal that is generated controlled? It's controlled by activation and deactivation by different inhibition pathways, and by phosphodiesterases, and we'll take an example at some. But here's the full picture of the adenylate cyclase signaling system. So what we talked about in terms of structure on the last few slides is protein kinase A, which has 2 regulatory and 2 catalytic subunits. We also talked about adenylate cyclase that binds ATP, and once it's activated, it converts those ATP molecules into cyclic AMP. We also talked about the heterotrimeric Gprotein and the receptor that binds to the signaling molecule. All right, so now we're going to take a look at a bigger picture of that adenylate cyclase signaling system. So if you have a hormone or a signaling molecule that binds to a receptor, right, and that receptor is a transmembrane protein that spans the entire lipid bilayer or plasma membrane. That signaling molecule, once it binds to its receptor, that will cause conformational changes within the receptor at really the cytoplasmic end, which will trigger activation of the G protein, right? Where now it will replace its bound GDP by GTP, thereby activating it. Once it's activated and its bound GDP is replaced by GTP, it will basically dissociate from its beta and gamma subunits. And then now you have this active alpha subunit of the heterotrimeric G protein bound to GTP, which will then bind to adenylate cyclase, right? And once it's bound to adenylate cyclase, it will activate it, right? It will activate adenylate cyclase by converting then, once it binds to adenylate cyclase, it activates it, and it causes it to synthesize cyclic AMP from its bound ATPs. Okay, 1 second. All right, so once the alpha subunit of the heterotrimeric G protein is bound to adenylate cyclase, it will stimulate adenylate cyclase to synthesize cyclic AMP from its bound ATPs. All right, and then those cyclic AMP molecules will bind to the regulatory subunit of protein kinase A, right? Once the very first cyclic AMP binds to the C-terminal end of the regulatory subunit, that will cause conformational change within the N-terminus domain of one of the regulatory subunit, allowing cyclic AMP to bind, and also allowing the catalytic subunit to dissociate from the regulatory subunit, right? So you have 2 cyclic AMP bounds per regulatory subunit. Now, the catalytic subunit can now interact with the substrate protein, phosphorylating it, right? So what it does, it causes or it triggers the phosphorylation of a number of different cellular proteins. Now, the magnitude of that signal, right, can be controlled by a number of different systems. Cyclic AMP can be hydrolyzed back or hydrolyzed into AMP through an enzyme known as AMP phosphodiesterase, which will in turn basically limit the signal, or it can't bind to phosphokinase A, so it can limit that signaling cascade. There's also proteins known as phosphoprotein phosphatase that catalyze the opposite reaction, or the dephosphorylation of specific proteins that can also limit that signaling cascade. Another one that you can have some toxins that can bind to the alpha subunit of the G protein, causing it to release its bound, causing the hydrolysis of its bound GTP, going back to GDP, or its inactive form. Well, it could also be that the signaling molecule itself was bound to an inhibitory, receptor protein, which then, the first set of events will still be the same, where it will cause the heterotrimeric G protein to displace its bound GDP by GTP, thereby activating it, and then it will separate the beta and the gamma subunit from the alpha subunit. But if it was bound, if it was an inhibitor external signal, it will not cause that alpha subunit of the heterotrimeric G protein will not bind to adenylate cyclase, or will not activate adenylate cyclase. Okay, so now when you think about that entire system of adenylate cyclase signaling system, make sure in the back of your head you could think about, the actual structure of either one of those proteins, like in the case of cyclic AMP, how it's specifically binding to the regulatory subunit, and then you have the autoinhibitory segment of the regulatory subunit at the N-terminus that will then dissociate from the catalytic subunit, thereby activating the protein. Okay, so a signaling molecule binds to the receptor undergoes conformational changes. It triggers the activation of heterotrimeric G protein, where now its alpha subunit will dissociate, will bind to adenylate cyclase, will activate it by, causing the synthesis of cyclic AMP from its bound ATP molecules, which can bound to to protein kinase A, thereby activating a number of different cellular proteins. And phosphodiesterases can limit the function of those secondary messengers, such as cyclic AMP, and we saw one example where cyclic AMP phosphodiesterases catalyzes the hydrolysis of cyclic AMP into AMP, right? And there are a number of different phosphodiesterases that can be distinguished based on which substrate they bind, their kinetic properties, their response to different inhibitors as well. Okay, and those phosphodiesterases are also targets to a number of different drugs, right? Ones that, they target different drug targets for heart failure, for asthma, for depression, also target those phosphodiesterases because they could limit the magnitude of the signal that a specific signaling molecule induces upon binding to its receptor. All right, I hope this was helpful.